Okay, continuing with topic 6, we were talking about all sorts of different organelles, and almost made it to where I wanted to talk about. Hadn't quite talked about the peroxisomes yet. Before we get into that, I do want to continue with the tradition here of talking a little bit about lab reports. I'm hoping everybody has done a little bit of work on your lab report. As I have warned you before, this is a big piece of work and worth 12% of your grade, and it is not going to be able to be doable in one night. So I've talked a little bit about results so far, a little bit about titles. Today I want to talk about introductions and methods. And those are actually probably some of the more easy sections, although it's going to take you a little bit of time to write these things. So let's talk about the introduction. The introduction is a written section, and it is going to have basically 3 things: background information, and objectives and hypotheses. And so this is about a page and a half. You could look at this as about 3 to 4 paragraphs, maybe 1 paragraph for each thing. The background information might be 2 paragraphs, kind of depending on what you're looking at. And by background information, it means we want you to set the context for the experiment. So, mention that this is an experiment about membranes. Maybe there will be a sentence or 2 about the importance of membranes, and then kind of get down into, the phospholipid bilayer or the fluid mosaic model, because that's going to be relevant to your objectives, because the objectives here are testing temperature and solvents on the integrity of these membranes. Maybe mention organism, that's beta vulgaris is the scientific name, and why we're using that particular organism. So that's going to be in there, and then your hypotheses are basically your educated guesses about what you think is going to happen. Of course, we've done the experiment already, so you probably know what's going to happen, but try to stick with your original hypothesis, what you thought it might have been, and then that's going to be discussed in the discussion part. So the other thing that you may do in the introduction or the discussion is you have to have 1 citation in your paper somewhere. It has to be in the introduction or the discussion, either. And we'll talk about citations. We'll talk about that maybe next day a little bit. Methods, I want to talk about that just a little bit more. The methods, you are not going to write out a huge detailed method. I don't want 5 pages of just you copying the lab manual. There's 2 things I want you to do. 1, I want you to cite the lab manual. So your very first sentence should be something like, the detailed procedure for this lab is found in the Biology 107 lab manual, and then the citation. So there's the citation. There's a hint on it. If you remember how to do them back from assignment 1, right? As you put the author. In this case, there's a bunch of authors and they're not listed in the lab manual, so you use Keano College, and then followed by the year, and that's all going to be in brackets. And the end of the sentence is after the brackets, so that's where the period is going to go. The second thing you're going to do is have a concise description of what was done. So you're not going to go and tell me everything, but I want you to talk about the variables and exactly what is done around those variables. I'll give you some examples here, an example here in the moment here on the next page. So take a look at this. If you think about our lab, there were basically 3 or 4 sections, right? We had the standard curve, the preparation of the beetroot discs, the temperature experiments, and the solvent experiments. So think about that as really 1 or 2 sentences for each section. So take a look at this. Here's something talking about the temperature part of the experiment, right? And there's that sentence, right? The very first sentence, right? About citing the lab manual. The second sentence, you can see 6 beetroot plugs were prepared. So that's actually part B, or maybe that's part C of the experiment. Part A is the standard curve, which is not in there. This, by the way, is actually directly right from another student. So this student missed the standard curve. But anyway, take a look at this. The beet plugs are mentioned there, and then the temperature experiment. And I'll talk about that in a second. Take a look at the second one, and I'll tell you which one I think is a lot better. So the second one, it says we have a ruler and razor blade were used to measure and cut the discs. The discs were placed in a cheesecloth, put in a plastic beaker, and all those kind of things. So the better one here is the first one. The second one has lots of good information, but those are not relevant details here. They're really relevant if I do want to reproduce the experiment, but that's where I'm going to go all the way to the Keanu lab manual, and I'm going to read those minute details. What I'm looking for is a concise description around the variables, right? So in this case here, telling me that some beet plugs were prepared is good enough. I don't need to know all the details about how exactly they were prepared, that there were 6 of them, right? Or actually, we made more than 6, so that actual number is wrong. So whoever did that one needs to fix that one. And exposed to various temperatures, right? So look at this, I'm even listing the temperatures. So there's the variable temperature, here's the actual temperatures, and in your experiment, we didn't actually include the 85, so you can cross that one out. And if any of the other temperatures were different, this is where you're going to list it. They're exposed for 2 minutes. So the temperature experiment is probably going to take maybe 2 sentences, maximum 3. And you're going to hopefully be able to do this in half a page. So like I said, it's very concise, just kind of giving the essential core details around the variables and the specific temperatures or the specific solvents and things like that. So it's going to take a bit of reading to read through that procedure and figure out a way to condense it all into half a page. As always, if you have any questions, do ask me about this. But that's it for lab reports for today. Okay, so back to our organelles. If you remember, I'm just going to go back to this this Word document. We were producing this these notes here, which were kind of a little bit of I guess, concise summary of what we talked about with the organelles. So we got the cytoplasm, we got the nucleus, and all the things in and around the nucleus, the ribosomes, the rough ER, the smooth ER, the vesicles, the Golgi body. And then we were starting to talk about these specialized organelles. So we have the lysosomes, of course, which are digestive compartments, and the vacuoles, which basically hold things. There's 3 different types of vacuoles: the food vacuoles, central vacuole in plants, and contractile vacuoles in Paramecium. So hopefully we can see some of these vacuoles this week in lab 5. And 3 organelles to talk about, like I said, all kind of specialized type of organelles. So the peroxisomes are next. So the peroxisomes are seen there on that micrograph on the right. And sometimes they have an enzyme in it. I can't remember the name of the enzyme that kind of crystallizes. So you can see the enzyme sort of forms this big sort of square type of shape. And this is how they were discovered to be a unique special organelle. And it's kind of hard to say a lot about peroxisomes without getting into so many specifics, because there's so many different types. There's different types in plants and animals, and there's some in your liver that are different from the ones in your muscle cells. And there's kind of 2 things we can say about them. One is they're oxidative organelles that break stuff down, okay? And that's kind of the key thing that I want you to know for this class, is that they oxidize things. That means they break things down, and specifically, they break down large and branched fatty acids. So that's part of our eating, right? Our consumption of fuel is breaking down fatty acids. They're called peroxisomes because sometimes in these oxidative processes, the hydrogen peroxide is made. So peroxisome, hydrogen peroxide, and that's kind of a byproduct of some of these reactions, and they're able to actually break that down. And that's kind of a big part of it. And so, like I said, it's not worth getting into this without kind of getting into a whole bunch of biochemistry, which we're not going to do in this class here. So let's make a note for that. So peroxisomes, they break down, we'll say break down / oxidize various compounds, we'll say. And like I said, it kind of depends on what tissue and what type of animal or plant cell you're in, right? And the example to know is they oxidize long or branched fatty acids. So if you eat any unusual fatty acids in your diet, normally normal fatty acids are broken down in the mitochondria during kind of a normal respiration type of process. But peroxisomes, they take care of the weird ones, the ones that are branched or extra long. So that's kind of mostly it for peroxisomes. I was going to show you one kind of extra one here, just sort of as a bonus, won't be on the test. This is a specialized type of peroxisome. It's called a glyoxazone. So this is found in the seeds of plants. So you might know that a seed has a lot of energy in it. It's pretty energy rich, and most of that energy is actually found in fat. And but what is needed is that the fat needs to be converted to carbohydrates such as glucose for the germination process. And these are done in peroxisomes, which are a specialized one called Glyoxazone. So like I said, there's a lot of weird ones out there depending on the species and the tissue and things like that that are kind of interesting. But too many weird examples to kind of generalize other than to say they break things down. Lots of them found in your liver, the peroxisomes as well. So let's talk about the last two, mitochondria and chloroplasts. So notice there's this note here. It says these are not part of the endomembrane system. So the endomembrane system, maybe what I will do is make a little note over here, right? Endomembrane system. Okay, so I'm going to make that separate here from the mitochondria and chloroplasts, and just make a note of what that includes, right? So we have the nucleus, or the nuclear membrane at least. The nucleus, we have the rough ER and the smooth ER. We have vesicles. We have the Golgi. We have the plasma membrane. We have lysosomes, peroxisomes, and vacuoles. Okay, so all of those organelles are part of the endomembrane system, and that means that they're connected, either directly connected, the membranes are directly touching each other, or they're connected in that vesicles can shuttle materials back and forth. The mitochondria and chloroplasts are different. They don't connect to the other organelles. They don't receive or send out vesicles between two other organelles. So they're kind of, a little bit unique anomalies here. The other thing is they kind of have unique membrane systems. If you take a look at the mitochondria, it's got those folds on the inside, and the chloroplast as well has those little pancake-shaped green things. We're going to talk about those in a moment. The other thing you may notice is that mitochondria and chloroplasts have their own DNA. So that's kind of interesting, right? So it's kind of worth talking about that for a minute here, but let's just take a look at a little bit of a better look at mitochondria and chloroplasts. And like I said, I want to highlight a few things, and we're going to come back to and talk about these things when we talk about respiration and photosynthesis, of course. So maybe actually that's a point we can make here is, I'm just going to go to my Word document for a minute. You probably know that mitochondria are the site of cellular respiration. So much more on that in a few topics. Topic maybe 10, I think. I can't remember exactly. And chloroplasts are the site of photosynthesis. And that's all we're going to say about kind of their functions for now. Like I said, we're going to get into a lot of details on respiration and photosynthesis in some later units, but nothing to worry about at the moment. That's for later. So that's kind of it for these notes, by the way. I do have another note to make about something else here in a minute. But I want to just take a quick look at mitochondria and chloroplast structures. So take a look at the mitochondria there. Notice it has two membranes. So it has an inner membrane and an outer membrane. So it's a double membrane organelle. It has its own DNA, and take a look at that. It's showing the DNA is actually a circular piece of DNA. So we're going to talk about that in a minute. You've seen circular DNA before. So the inner membrane kind of folds in on itself, and some people say the folds look like little fingers, and those were given a name many years ago by electron microscopists people, and so they were called cristae, which is a term I never use. I just say inner membrane because that's what it is. Some more pictures of some mitochondria. Here's one of those pictures that kind of has the electron micrograph fused with a cartoon to sort of show you what they think is going on. And you can see they look very bean shaped. It may not necessarily be bean shaped, but that's maybe a story for another time. There's another electron micrograph from a muscle cell, and often they're colored red. They're not necessarily red organelles, but red is, implying oxygen, which is usually also colored red. And of course, mitochondria use oxygen as part of cellular respiration. There's another one, lots of little cartoony pictures. You can see, again, very bean shaped, and you can see the inner folds and the inner membrane on that very nicely. And then there's another one. This is one of my favorites. I think it's just a very beautiful picture showing the inner folds. It's a scanning electron micrograph. Oh, and there's some more, lots of pictures of mitochondria. I don't get tired of pictures of mitochondria for some reason. So let's take a look at the chloroplast structure for a minute. And like I said, both of these things we're going to come back to when we talk about respiration and photosynthesis. Chloroplasts, of course, are the site of photosynthesis, and they actually have 3 membrane structures. So notice they have out here, that's the outer membrane, and then right here is the inner membrane, and then these green things right here are called thylakoid membranes or thylakoids, and they're green because that is where the chlorophyll is. The green part of the plant is the chlorophyll, and it's found right on those thylakoid membranes. So that's where the chlorophyll is. That's where the light is going to interact with the chloroplast, and of course, that's a big important part of photosynthesis. So much more in photosynthesis later. Notice it's also showing the DNA as being a circular type of DNA, and we'll talk about that in a minute. So we did look at chloroplasts in Elodea, and that was in lab, is that lab 2? Yeah, lab 2. And there's an image of some chloroplasts. And we're hopefully going to see some chloroplasts this week as well in, what are we looking at? Euglena as well. So looking forward to that. There's kind of an interesting fluorescence one. I don't exactly know what's going on in this algal cell in terms of the chloroplasts are sort of attached in this interesting network, but thought that was interesting. Something else to note about chloroplasts is often they store starch there, because of course starch is one of the products of photosynthesis. So where is it going to go? It's going to get stuck in the chloroplast. So I wanted to talk a little bit about chloroplasts and mitochondria in terms of why they're not part of that endomembrane system. We pointed out the fact that they both have they have DNA, and the DNA is circular. Maybe the other thing I didn't point out is probably in one of these pictures here. Yes, it is. They also have ribosomes. And so this is really unique. We have these organelles that have their own DNA and they have their own ribosomes. So in some ways they sound a little bit like they are their own cells. And they're not cells. We can't grow them alone in a petri dish by themselves. But the theory goes that maybe once in our deep evolutionary past, they were cells. And maybe the mitochondria were aerobic bacteria. Maybe the chloroplasts were aerobic, some sort of aerobic algae or some sort of aerobic photosynthetic algae or photosynthetic cyanobacteria or something like that. And so this hypothesis is called the endosymbiotic theory. And you can see it's kind of represented here, where you have some sort of ancestral cell, and it's taking in a mitochondria or chloroplast by endocytosis, and rather than killing it and eating it, it survived and has become the modern day mitochondria or chloroplast. So it's kind of an interesting theory, and I just wanted to write down some of the evidence that supports this theory. So evidence that supports. And I'm not going to write down the endosymbiotic theory because it's already written on there. And it's not a complete hypothesis. We don't have a time machine to really fully understand how this happened or may have happened. But it's very fascinating and something that if you're interested in mitochondria and chloroplasts, certainly is worth a read, because there's a lot of really interesting things about them. So what is some of the evidence? One is that they have circular genomes. Circular DNA genomes, and I'll put in brackets like bacteria. Now, this is very different than a bacterial genome in some ways. The E. coli genome has more than 4,000 genes. The human mitochondrial genome has only something like 37 genes. So not really a complete organism, not even anywhere close, at least at this point anyway. What else? Ribosomes. And these ribosomes are 70S ribosomes, which is in some ways, they are much more similar to bacterial ribosomes than they are to eukaryotic ribosomes. So kind of interesting. Another one is size. Their size is similar to bacteria. Similar to bacteria. And the other one is the double membranes. So the double membranes, they kind of suggest that maybe these things were taken in by endocytosis. Because remember that in endocytosis, what you're doing is wrapping a membrane around something. So the idea is that maybe this was something with one membrane, and now it's been wrapped with another one during this supposed event in deep evolutionary past or something like that. Anyway, like I said, it's not a perfect model, but there's a lot of people that are very interested in this kind of thing, and it's worth mentioning about mitochondria and chloroplasts. And I'm not going to say anything more about it, but kind of fascinating. Okay. What else can we say about eukaryotes and organelles? There are a lot of defects that people can have in our organelles. And there's a huge, huge list of all sorts of genetic defects that can lead to all sorts of different diseases. I'm not actually familiar with most of these diseases on this list in terms of how they exactly work. Cystic fibrosis is one I'm most familiar with, maybe, and you can see it's actually due to a mutation in a chloride transporter that is found in endoplasmic reticulum. And it leads to all sorts of secretory issues in a person's digestive tract and in their lungs and things like that. And anyway, mainly the point is that if our organelles aren't working properly, our cells may not work properly, which can lead to various types of human diseases. So there's a lot of interest in these things, obviously. So the other thing I want to do is kind of just talk about different types of eukaryotic cells, because of course, we can just say there's eukaryotes, but of course, animals are very different from plants and so on. So it's worth it to highlight some of the major differences between these things. Here's your typical animal cell. And obviously these things are very cartoony, but I do want to point out some of kind of the big features around animal cells. Here's the one from the textbook. Again, I find these things a little cartoony, and it's kind of implying that animal cells all have flagella. That's the tail-like structure on there. And the reality is, no, not all of our cells have flagella or microvilli or anything like that. But we do have microvilli in our intestine. We do have flagella in our sperm cells and so on. And these are features that aren't necessarily generally seen in plant cells. I'm saying generally because there are exceptions of plant cells that have sperm, that are kind of like sperm, but I'm not going to get into all that. You can learn a little bit about that in Biology 108. But it's showing a bunch of features here, and if you kind of take a look at what an animal cell is, there's a couple of things we've talked about already, right? So one, of course, is that animal cells do not have a cell wall. Right? So that was something we talked a lot about with animal cells. They may have an extracellular matrix, right? Remember that some animal cells may have an extracellular matrix, which includes collagen and connective tissue and bone tissue and things like that, but not all animal cells have that. Another thing to say about animal cells is that they are chemoheterotrophic. You're probably looking at that word and wondering what it means. I know it is taught in high school. And basically it means you're feeding by ingestion, right? So chemo means a chemical, hetero means other, and trophic means feeding. So it means you're eating another organism. That's what it means. That's how we get our energy. We feed by ingestion. So humans, all animals feed on different things, right? And you may feed on plants, you may feed on animals, you may be an omnivore, you may feed on both things. So those are two kind of big things we can say about animal cells. We can say a lot of other things about animals. Animals in terms of, their classification. We tend to be mobile, maybe meaning we move around, but not all animals do. Sponges are animals and they don't move around, not generally. Maybe their larval stage does. I'm not sure. I don't know a lot about sponges. Animal cells tend to have centrosomes and centrioles and things like that. And there's a lot of kind of little particulars we can say about them, but those are kind of the two big things I want you to know about animal cells, at least for now. There's some electron micrographs down there. You can see these are intestinal cells, and these little structures here are actually called microvilli. And the microvilli are there to give the intestinal cells extra surface area for digestive process. You can see the nucleus and the nucleolus quite visible in these animal cells, and they look very nice. A lot of this roughage that you're seeing there in the middle, that's actually endoplasmic reticulum, and depending on the image, sometimes it comes out really nice. In this case, it's kind of just looks like a bunch of roughage, but that's what most of that stuff is. It takes up a massive part of the cell. Here's another animal cell, another colorized electron micrograph. You can see it actually looks like this one here has 2 nucleoli, which is not uncommon in animal cells. Some plant cells can have up to hundreds, which is kind of nuts when you think about it. But typical for animal cells to have 1 or 2, I don't know what the plural is, nucleoluses. I'm not sure if that's the plural or not. These red things all over here, pretty much everywhere in this cell, these red things are mitochondria. So this is loaded with mitochondria. And the little blue spots are ribosomes, and some of this black stuff might be some endoplasmic reticulum here that they've labeled and colored. The yellow things are lysosomes. So it's nice when somebody's colored it for you and told you what everything is in there. But you can see no cell wall. And this one here is probably a muscle cell. Oh no, it's a liver cell, it says, which means it's going to probably consume a lot of energy, which is why it has a whole bunch of mitochondria. So what about plant cells? There's the picture from the textbook, and you can see that right away is very different. First thing to notice, it has a cell wall, right? So cell wall, that's one big difference here. Number 2 is it has chloroplasts. So chloroplasts, those are kind of the 2 big things to notice. They do often have those central vacuoles. I'm not going to focus on that too much for water storage. Some plants or some animal cells have vacuoles for water storage, but plant cells is really, really common to have a big central vacuole for water storage. We could talk about how there's no centrioles here. There, like I said, there's chances are you have more nucleoli and whatnot, but these are kind of the 2 things to mention here. They have a cell wall and they have chloroplasts. So the cell wall in plants, of course, is made out of cellulose, and these things are self-feeders. So we call these ones here photoautotrophs, which I thought I had that word on here. So photo means light, auto means self, and troph means feeding. So they're self-feeders. They make their own food, and they make it from photons, which photons, which is of course light. And you know that about plants, I'm sure you do. And that's kind of the 2 big things to know about plants, right? Cell walls and their photoautotrophs. There's some duckweed there. I don't know if anyone here has ever had duckweed in their aquarium. It's kind of nuts. You get it in your aquarium and it just grows like crazy all over the place. I remember having some in my aquarium once. It was hard to get rid of. You can see all the green chloroplasts, and these orange things here are mitochondria. Like I said, they're usually colored red, but orange in this one. You can see there's the nucleus and nucleolus there. And so very nice pictures of some plant cells there. There's another plant cell. In this case here, the chloroplasts are actually colored in yellow. So this is a chloroplast right here. Not sure why they didn't make them green, but like I said, the colors are not accurate in an electron micrograph. There's the nucleus, big nucleus in the middle with the nucleolus, lots of mitochondria. I don't think they have any peroxisomes or lysosomes colored in there. And then you can see the cell wall around on the outside. So this is kind of an unusual plant cell because usually they're kind of more box shaped because of their cell wall. So I'm not sure what species this is from. maybe you can click on the link and check it out. Okay, a couple more to mention. Number 3 are the fungi. So remember, the fungi also have cell walls, and these cell walls are made out of chitin. So chitin is different than cellulose. It's similar. It's actually a similar carbohydrate made out of N-acetylglucosamine instead of made out of glucose. And it's kind of doing the same thing. It's forming a structure. Fungal cells can include, so if you're not as familiar with fungi, some people are not. So fungi include things like yeast, mushrooms, molds, puffballs. There's a lot of weird things out there, but those are kind of the big 3 forms that you would see of fungal cells. And they are also chemoheterotrophs. So, whoa, that's the same word we used for animal cells. They're chemoheterotrophs. That means they eat other organisms. The difference is that fungal cells don't walk around and eat things. They're usually decomposers, meaning that they don't have stomachs, but they secrete their enzymes instead. But in some ways, fungal cells are more similar to animal cells. And on the phylogenetic tree, they're much closer to animal cells than plant cells. So no photosynthesis here, okay? Do not get these mixed up with plants. Fungi and plants are very, very different organisms in many, many ways. Notice that this one here has a vacuole, and you can see the nucleus and the mitochondria. And I think a lot of this dark stuff here is endoplasmic reticulum, would be my guess. It's not labeled on the micrograph. There's another fungal cell here in the middle. And I was looking for a little diagram that was talking about protists. And so remember what a protist is. So a protist is basically, it's a eukaryote that is not an animal, plant, or fungi. The other thing about protists we can say is that they're usually unicellular, but not always. There are actually quite a number of cases of protists, and sometimes, maybe the last note I'll say about protists is sometimes they're grouped with algae. Sometimes algae is its own group. Sometimes algae is grouped with protists. You can see that definition of protist basically meaning it's a eukaryote. It has a nucleus, and it's not a plant, an animal, or a fungi. So that would include algae, because algae are not plants. Algae are plant-like organisms in that they do photosynthesis, but they're usually unicellular. Like plants, they have vascular systems and tissues and things like that, whereas algae are very simple organisms. And there's some weird algae out there. You can see this one here on the left, this algal cell here on the left. It doesn't say the name on it, but this cell has flagella, so it can swim around. So that's very different than your typical kind of plant cell. So some of these are really weird organisms. They're animal-like in that they swim around, and they're plant-like in that they're photosynthetic. So that's why they're grouped into this whole big thing called protists, which, if you think about kingdoms, protists include many, many kingdoms. And in fact, biologists are trying to get away from that concept of kingdom nowadays because it just kind of gets muddled and confusing. So in terms of a protist, I want you to know what they are in terms of that definition I have there. And know that there's a huge variety. Some are photosynthetic, some are not. Some have flagella, some do not. I'll show you the protists that we're going to be looking at in this week's lab here in a moment. This one here is from the textbook. This is one we could look at, but we're not getting this one this year. It's called how do I say that? Chlamydomonas, I guess, if I'm saying that right. So you can see this one here is it's photosynthetic and it can swim around. So kind of animal-like and kind of plant-like at the same time. So you can see my note there. It says many kingdoms, most are unicellular. So let's take a look at the ones that we're going to try to look at this week. We're going to look at the Paramecium. So Paramecium looks like this. You can see there's a micrograph on the right and the cartoon on the left. You can see it's kind of interesting. It has 2 nuclei. So notice it has something called a macronucleus, which is a big one, and a micronucleus, which is a small one. So kind of interesting there. I'm hoping you'll be able to see the macronucleus when we look at them under the microscope. The other thing, if we're lucky, we'll be able to see is the contractile vacuole. That was something we've talked about in class as something that pumps out the water. And sometimes we can see these cilia. Kind of depends on the condition of our paramecium. Sometimes they're kind of, sometimes they're really zippy and energetic and they move too fast. And then other times they're kind of dead. So we want to have them somewhere in the middle, and hopefully we can control for that in the lab. Again, sometimes it depends on the shipping, and particularly in February, sometimes we get out of luck with the shipping, things stuck in trucks too long and whatnot. But there's the paramecium. Another one we're going to look at is the Euglena. And so the Euglena is photosynthetic, and it has a flagellum. So the flagellum is the structure here for swimming around. And so these ones here, sometimes it's hard to make out most of the structures. Sometimes you can make out the nucleus, and sometimes you can make out this eye spot. So the eye spot is a little organelle that senses light and allows it to swim towards light, because of course this is photosynthetic, and that's where it wants to go. It wants to swim towards light in order to do its photosynthesis. The other one that we're going to look at is this one here. It is called an amoeba. The one in particular that we usually look at is called a Pelomyxa, and you can see I actually took that photograph on the right there, and this was the one that the other two we usually get a chance to look at. This one here sometimes they tend to be really fragile. Last semester, I think we're out of luck. Last winter I had some live ones. I would say we have a 50-50 chance of having some live ones, unfortunately. But if we're lucky, we'll get to see these, and they're really fascinating to watch them. They move around and they change the shape of their membrane, and it's very cool. You can see these projections here are called pseudopodia. I think we talked about this before. So pseudopodia, pseudo means fake or false, and podia means feet. And these are the projections of the membrane changing its shape. So this week's lab is short and fun, and it's pretty much ideal for this week, considering it's right before the midterm. Okay, just looking at a time check here and see where we are with everything. Uh-oh, what's my PowerPoint doing? It just gave me a weird error message. I don't know if you guys saw that. Now it can't go. There we go. What's going on here? Guess I clicked my button too many times. I do have these 2 little videos here. I'm going to see if I can play them for you. And they're 3 minutes long. They kind of give a quick tour of the cell and a good recap of these things here. So let me see if I can play them for you. And all right, oops. Let's give that a try. Here we go. Okay, maybe not. I may have to find the original video file. So just give me a second here. You know, my videos were working last year, and this year they don't seem to be working so well. All right, just give me a second here. Okay, there they are. I found the video files, and I'm just going to play them here in a second. All right, so here goes the one on the animal cells. Hold on a second. I'll just share my, oh, where are they? Come on now. Oh, come on. Ah, I'm sorry about this. Try that. Okay. Just a second. Let me just exit from the PowerPoint. I don't know why these files are doing this to me. Fun. Yeah, for some reason I have them loaded up, but Zoom can't find these video files. So I have one other options I can use here, and what I do is there are some links for YouTube of these things here. So I'm gonna do that. Just go to YouTube. So there we go. Let's try that. Hopefully,
students with ADHD save 10 hours studying with the
too
screen
many
extension.
ads.
But no.
All right, here we go with the animal cells. Sorry about that.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The Endoplasmic Reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Cooking is about using your senses and intuition.
Okay, so I hope that that was good. I'm going to now play the one for the plant cells. And this was a little bit different because plant cells, of course, are very different. So let me just get that up and going now.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. All plant cells have mitochondria, just like animal cells do. Sugars produced by photosynthesis are broken down and converted to ATP in mitochondria. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
Okay, so that's actually it for topic 6. On Wednesday, we are going to be doing a review session for midterm number 1. So don't forget about midterm number 1. That's on Friday. I'll be sending out some instructions over the next day or two. I was just checking, and only one person has done the sample quiz, the lockdown browser sample quiz on Moodle. It's really important you do that. I'm not responsible for if your technology does not work out. We would like to do any troubleshooting ahead of time. So it's important that you go and do that. I will be sending people an email reminder. I should not have to do this. I should not have to send you an email reminder. So please do take a look at that as soon as possible. And I will see everybody on Wednesday. So have a great day.